4-hydroxybenzoic acid. 4-hydroxybenzoic acid, also known as p-hydroxybenzoic acid (PHBA), is a monohydroxybenzoic acid, a phenolic derivative of benzoic acid. It is a white crystalline solid that is slightly soluble in water and chloroform but more soluble in polar organic solvents, such as alcohols and acetone. 4-hydroxybenzoic acid is primarily known as the basis for the preparation of its esters, known as parabens, which are used as preservatives in cosmetics and some ophthalmic solutions. It is isomeric with 2-hydroxybenzoic acid, known as salicylic acid, a precursor to aspirin, and with 3-hydroxybenzoic acid. Natural occurrences. It is found in plants of the genus Vitex such as V. agnes castus or V. negundo, and in Hypericum perforatum St. John's wort. It is also found in Spongiachlori spongiosa, a freshwater green alga. The compound is also found in Ganoderma lucidum, a medicinal mushroom with the longest record of use. Cryptoneribacter phenolicus is a bacterium species that produces benzoate from phenol via 4-hydroxybenzoate. Occurrences in food. 4-hydroxybenzoic acid can be found naturally in coconut. It is one of the main catechins, metabolites found in humans after consumption of green tea infusions. It is also found in wine, in vanilla, in Macrotyloma uniflorum horse gram, carob, and in Philanthus acidus otahate gooseberry. Acai oil, obtained from the fruit of the acai palm Euterpe oleracea, is rich in p-hydroxybenzoic acid 892 plus 52 mg/kg. It is also found in cloudy olive oil and in the edible mushroom Russula virescens green cracking russula. Related compounds. P-hydroxybenzoic acid glucoside can be found in mycorrhizal and non-mycorrhizal roots of Norway spruces Picea bies. Vialdilphin is an anthocyanin, a type of plant pigments, found in blue flowers and incorporating 2 p-hydroxybenzoic acid residues, 1 rutinoside, and 2 glucosides associated with a delphinidin. Agneuside is the ester of aucubin and p-hydroxybenzoic acid. Metabolism. Biosynthesis. Chorusmate lyase is an enzyme that transforms chorusmate into 4-hydroxybenzoate and pyruvate. This enzyme catalyzes the first step in ubiquinone biosynthesis in Escherichia coli and other gram-negative bacteria. Benzoate 4-monooxygenase is an enzyme that utilizes benzoate, NAD, H+, and OTWO to produce 4-hydroxybenzoate, NAD+, and H2O. This enzyme can be found in Aspergillus niger. As an intermediate, the enzyme 4-methoxybenzoate monooxygenase or demethylating transforms 4-methoxybenzoate and electron acceptor AH2 and OTWO into 4-hydroxybenzoate formaldehyde, the reduction product and H2O. This enzyme participates in 24-dechlorobenzoate degradation in Pseudomonas putida. The enzyme 4-hydroxybenzaldehyde dehydrogenase uses 4-hydroxybenzaldehyde, NAD+, and H2O to produce 4-hydroxybenzoate, NAD, and H+. This enzyme participates in toluene and xylene degradation in bacteria such as Pseudomonas mendocina. It is also found in carrots Daucus carota. The enzyme that 24-dihydroxyacetophenone dioxygenase transforms 24-dihydroxyacetophenone and OTWO into 4-hydroxybenzoate and formate. This enzyme participates in bisphenol degradation. It can be found in alkali gene species. The enzyme 4-chlorobenzoate dehalogenase uses 4-chlorobenzoate and H2O to produce 4-hydroxybenzoate and chloride. It can be found in Pseudomonas species. The enzyme 4-hydroxybenzoyl-CoA thioesterase utilizes 4-hydroxybenzoyl-CoA and H2O to produce 4-hydroxybenzoate and CoA. This enzyme participates in 24-dechlorobenzoate degradation. It can be found in Pseudomonas species. The enzyme 4-hydroxybenzoate polyprenyltransferase uses a polyprenyl diphosphate and 4-hydroxybenzoate to produce diphosphate and 4-hydroxyminus 3-polyprenylbenzoate. This enzyme participates in ubiquinone biosynthesis. The enzyme 4-hydroxybenzoic geranyl utilizes geranyl diphosphate and 4-hydroxybenzoic to produce 3-geranyl-4-hydroxybenzoic and diphosphate. Biosynthetically, alkanin is produced in plants from the intermediates 4-hydroxybenzoic acid and geranyl pyrophosphate. This enzyme is involved in shikinin biosynthesis. It can be found in Lithosphaermum erythrohyzin. The enzyme 3-hydroxybenzoic CoA ligase uses ATP, 3-hydroxybenzoic and CoA to produce AMP diphosphate and 3-hydroxybenzoyl CoA. The enzyme works equally well with 4-hydroxybenzoic. It can be found in Thara aromatica. Biodegradation. The enzyme 4-hydroxybenzoic 1-hydroxylase transforms 4-hydroxybenzoic NAD2H+ and OTWO into hydroquinone NAD+, H2O and C-OTWO. This enzyme participates in 24-dechlorobenzoic degradation. It can be found in Candida parapsilosis. The enzyme 4-hydroxybenzoic 3-monooxygenase transforms 4-hydroxybenzoic NAD H+ and OTWO into protocatechate NAD+ and H2O. This enzyme participates in benzoic degradation via hydroxylation and 24-dechlorobenzoic degradation. It can be found in Pseudomonas putida and Pseudomonas fluorescens. The enzyme 4-hydroxybenzoic 3-monooxygenase NAD utilizes 4-hydroxybenzoic NAD H+ and OTWO to produce 34-dihydroxybenzoic protocatechuic acid NAD+, and H2O. This enzyme participates in benzoic degradation via hydroxylation and 24-dechlorobenzoic degradation. It can be found in Corynebacterium cyclohexanicum and in Pseudomonas sp. The enzyme 4-hydroxybenzoic decarboxylase uses 4-hydroxybenzoic to produce phenol and C-OTWO. This enzyme participates in benzoic degradation via coenzyme CoA ligation. It can be found in Klebsiella aerogenes, Aerobacter aerogenes. The enzyme 4-hydroxybenzoic CoA ligase transforms ATP, 4-hydroxybenzoic and CoA to produce AMP diphosphate and 4-hydroxybenzoyl CoA. This enzyme participates in benzoic degradation via CoA ligation. It can be found in Rhodopseudomonas palustris. Coniacidia hoffmanniae is a plant pathogen that commonly inhabits fertile soil. It is known to metabolize aromatic compounds of low molecular weight, such as p-hydroxybenzoic acid. Glycosylation. The enzyme 4-hydroxybenzoic 4-O-beta-D-glucosyltransferase transforms UDP glucose and 4-hydroxybenzoic into UDP and 4-beta-D-glucosyloxybenzoic. It can be found in the pollen of Pinus densiflora. Chemistry. The Hammett equation describes a linear free energy relationship relating reaction rates and equilibrium constants for many reactions involving benzoic acid derivatives with meta and para substituents. Chemical production. 4-hydroxybenzoic acid is produced commercially from potassium phenoxide and carbon dioxide in the Kolb-Schmidt reaction. It can also be produced in the laboratory by heating potassium salicylate with potassium carbonate to 240 deg C, followed by treating with acid. Chemical reactions. 4-hydroxybenzoic acid has about 1/10 the acidity of benzoic acid, having an acid dissociation constant Ca equals 3.3 m at 19 deg C. Its acid dissociation follows this equation. HOC6H4C OTWO H equals HOC6H4CO2 plus H plus. Chemical use. Vectran is a manufactured fiber, spun from a liquid crystal polymer. Chemically, it is an aromatic polyester produced by the polycondensation of 4-hydroxybenzoic acid and 6-hydroxynethyl 2-carboxylic acid. The fiber has been shown to exhibit strong radiation shielding used by Bigelow Aerospace and produced by Stemrad. 44-dihydroxybenzophenone is generally prepared by the rearrangement of p-hydroxyphenolbenzoate. Alternatively, p-hydroxybenzoic acid can be converted to p-acetoxybenzoyl chloride. This acid chloride reacts with phenol to give, after decetylation, 44-dihydroxybenzophenone. Examples of drugs made from PHBA include nifurexazide, orthocaine, ormeloxifene, and proximeticaine. Bioactivity and safety. 4-hydroxybenzoic acid is a popular antioxidant in part because of its low toxicity. The LD50 is 2200 mg/kg in mice oral. 4-hydroxybenzoic acid has estrogenic activity both in vitro and in vivo and stimulates the growth of human breast cancer cell lines. It is a common metabolite of paraben esters, such as methylparaben. The compound is a relatively weak estrogen, but can produce uterotrophy with sufficient doses to an equivalent extent relative to estradiol, which is unusual for a weakly estrogenic compound and indicates that it may be a full agonist of the estrogen receptor with relatively low binding affinity for the receptor. It is about 0.2% to 1% as potent as an estrogen as estradiol.